Hey everyone, welcome back to another video by Camroom 5. Today we are discussing the experimental study for osmosis. We've already talked about what diffusion and osmosis are, so why not let's discuss some aspects of it in experimental way. For that we need potato and some pieces of it. Then we need 5 different test tubes. We're going to be labeling them A, B, C, D, E. Each has a different concentration of a salt. Let's suppose A contains 0.02 mole per decimeter cube concentration, B has 0.04, C got 0.06, D got 0.08 mole per decimeter cube, and then E has 0.10 mole per decimeter cube. Now we put the pieces of potato in each test tube. Potato is a living cell and it has a cell membrane, right? We measured the mass of potato strips before we put them inside the test tube and after given time. In the beginning, each potato strip was identical, and they all had hypothetically the same mass as like 10.00 grams. But after, let's say, 10 minutes, the mass is changed. You can see even the shapes in the diagram has changed. In test tube A, it became more heavier. In test tube B, it became slightly little heavier, slightly little more massive. In C, D, and E, you can see the final mass is lesser than the initial mass. The explanation is that in test tube A and B, water moved inside the potato strip from the test tube. But in test tube C, D, and E, water moved outside. In test tube A, the most water moved inside from the test tube solution inside the potato, and in test tube E, most water moved from the potato outside. Why did that happen? The reason is that in test tube A, the water potential of potato was lower compared to test tube A solution. And that is why the test tube A solution was higher water potential, which means solution A was less concentrated, so it had more water potential, and then water moved inside. In test tube E, the water potential of potato was higher compared to the test tube E solution itself. The reason is that solution E was immensely concentrated, which means it had low water potential, so potato donated some water. If we make a graph about water moving in and water moving outside on the y-axis and concentration on the x-axis, we can see there's a change in mass for each potato strip. But sometimes there's an increase in mass and sometimes there's a decrease in mass. Test tube A has almost 6 units increase. When we talk about test tube B, it had almost 1 unit mass increase. Test tube C, D, and E did not have masses increasing, but they had a decrease in mass. Test tube D had almost 4 units decrease in mass, and test tube E had almost 7 units decrease in mass. That's why the mass dropped from 10 units to almost 3 unit grams. If we draw a line to connect these points like the best fit line, we will be able to see that when the solutions were very less concentrated, like A and B, the concentration was low compared to the potato itself. And then we can see the masses decreased because C, D, and E had a higher concentration compared to the potato. The question is, what's the concentration of potato itself? So it is when the line touches 0. 0 means that the amount of water coming inside was cancelled by the amount of water moving outside. the water potential of potato cell is equal to the water potential of outside solution. We can see this graph tells us this is almost a value of 0.48 something, 0.048 rather, or you can say 0.05. This is because the concentration of potato cell is equal to this value where it touches 0, so it is almost 0.44, 0.45 something. I should make it 0.04 something, but you can get the idea. Moving on, let's do another experiment where we will again study osmosis. Imagine two beakers, beaker X and beaker Y. Both beakers have two differently concentrated solutions. Beaker X has a concentration of 0.050 mole per decimeter cube. Beaker Y has a concentration of 0.0500 mole per decimeter cube. It means Y is 10 times more concentrated than X. X is very dilute, less concentrated, which means more water potential. Y, on the other hand, is more concentrated. It has less water potential compared to X. In both cases, I have inserted a tube which is basically a Thistle funnel. It is inverted and it has a semi-permeable membrane fixed on the surface. It is inverted and inserted in a way that you can see that there is some liquid inside both the tubes to the same level. If we use a ruler and we measure the amount of liquid level, the level of liquid, we can see that it is same in both cases. Level of liquid at start. And when we see it is same, X and Y both have almost 5.30 centimeters. It's a random value. But after a while, you can see that the level of liquid in X increased and the level of liquid in Y decreased. The level of liquid at the end of the experiment was changed for X and Y. In X, it became 10.00 centimeters, which means it increased. And in Y, it became 2.50 centimeter, which means it decreased. The explanation is that water moved inside the tube from beaker X because beaker X had high water potential, but in beaker Y, the water moved outside the tube because beaker Y was lower water potential. The reason is again osmosis, because osmosis means water moving inside from higher water potential to lower water potential. So liquid was lower water potential than X, but liquid was higher water potential than Y. I hope these ideas are clear to you guys. In the coming videos, we'll talk about some past paper questions also. Stay tuned guys, thanks.